Hi all, Dr. Clark here again, and today for animal biology I'm going to discuss Hardy-Weinberg and population genetics. It'll kind of just be a brief overview of the concepts of population genetics, how it kind of works, and then we'll go into more detail about ways in which populations can evolve, and a little bit more detail on, today population genetic studies of modern times. Okay, but before we get to that, I kind of want to talk about these two individuals, which are known by Hardy and Weinberg. Okay, this is Wilhelm Weinberg, and this is Godfrey Hardy. Okay, and they were, you could say, like precursors to statisticians. Okay, in 1908 is when they came up with what we call Hardy-Weinberg equilibrium. But they were interested in Darwin's evolution by means of natural selection and how species can change. And what they're really interested in is determining whether or not a population is evolving. Okay? So they devised a method to analyze the frequency of alleles in a population by just examining phenotypes. Now, this is based purely on Mendelian genetics and the fact that you're going to have a one-to-one relationship between the phenotype and the genotype. So this is just like simple Mendelian genetics. There's no codominance, no incomplete dominance, no epistasis, no environmental gene-by-gene interaction. None of that stuff is going on. Just simple genetics based on a single gene giving a single phenotype. Okay? So we're going to talk more about Hardy-Weinberg after I kind of introduce population genetics. So populations, as most of you know and probably have heard definitions of them before, are a group of organisms that can reproduce together. They occupy the same place, same time, okay? And therefore, when we're talking about population genetics, they also have to share a set of genes, which is normally what we call the gene pool. So they share the entire gene pool or all the possible alleles for that given population. Okay? And for some populations, their population is the entire species. So, for example, like devil's hole pupfish, you've heard me talk about them before. They occupy one region, one place on the planet. That's the entire population. That's the entire species. Now, if you talk about, say, humans, we might have many different populations, okay, based on the region of the world, okay, but the species occupy all those places on the planet. Okay? And so to examine the gene pool of humans, you would have to take samples of all the alleles from all the different populations throughout the world. It's very difficult. There's 7.7 billion people. There's, I don't know how many different populations, probably, well, of course, billions of populations out there. Okay? And so it's a very difficult point of view. Okay? So we'll talk more about individuals that are population geneticists, and how they get around not having to sample every single population, but still getting at whether or not a population is actually evolving. Okay, so often when we talk about population geneticists or population genetics, okay, we're going to talk about it from a frequency of alleles for a given characteristic. So if there's a phenotype in which you can easily observe coat color, flower color, whatever it might be. If we're talking about animals, maybe it's the coat, maybe it's the color of their fins, their feathers, the size of their antlers. There's lots and lots of variables, but for the most part, when population geneticists are trying to examine frequency of alleles, they're looking at single characteristics. Under Hardy-Weinberg, those characteristics have to be like traits that you could visualize, that you could actually phenotypically distinguish between one group and another. Modern geneticists, modern population genetics, has nothing to do with phenotype, because now we have the capability to examine genotype. Okay? And that's what a lot of modern population geneticists are doing. They're just examining genotype, and they really, in some cases, they really have no clue what gene, okay, is actually coding for the phenotype, or whether or not there is a phenotypic difference, okay, or physiological difference. Is there a behavioral difference? A lot of times they don't know. Okay? So it's kind of two different camps, and we'll talk about both ways at which this can be done. So again, bringing it back to gene pools are just the summation of all the alleles in a given population. Right? Now, like I said before, often gene pools are incomplete. We don't actually know how many different genes or how many different varieties of those genes are there for a given population. So you're studying, say, maybe you're studying a population of elk, okay, and you're examining what we call Rocky Mountain elk, and then you're examining maybe Pacific Northwest elk. Okay, now these are same species. Okay, depends on who you talk to. It might be different subspecies, but nonetheless, they're same species. They occupy different regions. They're two different populations. But I mean, if you're talking about Rocky Mountain elk, maybe there's 300,000, 500,000, somewhere in that range. Rocky Mountain elk between like Montana, Wyoming, Idaho, Colorado. There might even be more than that. There could be close to 1 million elk in that region. There's no way you're going to know every single different allele that's found in those organisms. Okay? So, but is it enough to account for 10% of the alleles, 20%, 30%, 40%? And so we'll talk about this as we progress. We'll look at, allele variation, and as long as you're examining the same alleles or same genes in one population and another, then it really doesn't matter how many different genes there are. And we'll talk about that also. Okay, so the gene pool really just represents how much variation or heredity is available. Okay, so it's the heredity resources. So what is possible in the given population? That's important to know in order to figure out whether or not that population is changing over time. Okay, so there are some sources of variation within a population. Some of these you're probably very familiar with because you learned about them in general biology. And so I'm going to kind of go through a bunch of ways at which the variation in gene pools can increase or decrease and change over time. Okay. First is independent assortment. Okay. So if you remember from your beginning biology course, you should have learned about something called independent assortment, in which chromosomes from the paternal line or the father line, chromosomes from the maternal line, the mother line, they pair up. Right? They line up on what we call the metaphase plate or the equatorial line, the center of the cell, during what we call metaphase 1 in meiosis. Right? So it looks like this. Now, how they line up is independent of one another. So it could be, let's say that red is representing maternal, blue is representing paternal. Okay, so dad's line, mom line, chromosome number 1, chromosome number 2, and this would go on and on and on for however many chromosomes the organism have. In our case, there would be 23. Okay. Now, how they pair up, whether chromosome number 1 is on the right side or the left side, is purely random. Okay, so here you can see the bottom chromosomes have switched orientation, okay, which would give you different variety of end product, okay, or gamete cells would be different genetically speaking. That's independent assortment. To figure out how much or how many variations you have, you can just take the number of chromosomes an organism has, okay, and whether they're got 2 copies of each one of those chromosomes or 10 copies of each one of those chromosomes, whatever it is, you raise the number of copies. So if they're a diploid, it's 2. If they're a triploid, it's 3, tetraploid 4, quintuploid 5, on and on and on, hexaploid, blah, blah, blah. Okay? You take that number and raise it to the number of chromosomes. Okay? So it goes to the power of number of chromosomes. In our case, it's 23. So 2 to the 23rd power. That gives you the amount of variation that you could expect from independent assortment alone. 2 to the 23rd power is, over 8 million. Okay, so just independent assortment from human point of view, we have 8 million different varieties that we could expect in the gametes just from rearrangement of chromosomes or the arrangement of chromosomes. Okay, the next one is crossing over. Also should be fairly familiar with you. In the case of your general biology course, should have talked about this. Crossing over happens when these homologous chromosomes, dad's chromosome and mom's chromosome, line up during prophase. So right here, right before this phase, this metaphase phase, they're going to be pairing up in prophase. And when they pair up, they have the capability to exchange arms or chunks of chromosome. Okay? And so dad's line, mom's line, okay, they can exchange material, which makes it neither dad's line nor mom's line, and it makes it what we call a hybrid chromosome. Now the amount of variation or the amount of possibilities for this to occur are basically endless. We don't even have the capability of predicting when this can occur. It occurs often enough that majority of the time when your gamete cells go through meiosis, majority of the time you will have multiple events of crossing over, multiple events of exchange of chromosomes. Okay? Now, does that mean every one of your chromosomes is going to exchange material? Nope. Does that mean that none of them are going to exchange material? It's possible. So that's what makes crossing over so interesting, is we don't know. And we don't know how much is going to cross over. We don't know where it's going to cross over. It doesn't have to just cross over at the tips. It could cross over in the middle. It could cross over on both arms. It's normally the inside arms of the chromosome that are crossing over. So the outside arms are untouched, not recombinated, but the insides are normally recombinated chromosomes or chromatids and result in this hybrid, both paternal maternal chromosomes. Okay? So another source of variation. Okay? Rearrangement of the number or structure of chromosomes. Now this one you may or may not have learned about in general biology, and that is when chromosomes become either stuck together or they rearrange themselves, okay, through mutations. So instead of switching material between 2 chromosomes, you're switching material within a chromosome. Okay? So like this side of the chromosome, the Q region could just flip over. So maybe it's Q2, Q3, and then it flips, and then it would be read Q1, Q3, Q2. Okay, that can occur. Okay? And some of the greatest examples of this have to do with human evolution. For a long time, human evolution becomes subject to a lot of criticism because there wasn't all that much data originally, and most of the data came from the fossil record, which fossil record is great, but because you only find a small number of fossil humans, it's sometimes difficult to put pieces together. Okay? Now remember, fossils are kind of spotty since you have to have the right amount of pressure, you have to have the right kind of soil types and the right sediment coming on top of them, you have to have the right amount of heat in order for bone to form into rock. You have to have all these features which are very unique okay, in order for that to occur. We know that human populations, at least prehistoric human populations, were fairly small. So the odds that you get a bunch of fossilized humans is pretty poor. And that's the case. So for a long time, scientists said, well, why can't we just look at this from a different avenue? We have ancestors that are currently alive, okay, through the great apes. So orangutans, gorillas, and chimpanzees are the other great apes. So we started examining, blood chemistry, protein structures, these kind of things, and noticed that they're nearly identical, but there was one problem. And that is when you look at the other great apes. The other great apes have 48 chromosomes. Humans have 46 chromosomes. And so for a while, there was, really until the early 2000s, there was this idea that, well, if humans belong to the great apes, why do they differ in the number of chromosomes? And so it made a kind of a test-the-wall hypothesis out there that indeed, if humans belong to the great apes, then somewhere in the human genome, somewhere in those 23 different chromosomes, you should find a chromosome that's a result of two chromosomes sticking together. And if you examine our chromosomes, in other words, what you should find is you have orangutans, chromosomes here, gorillas, chimpanzees, okay, they're all nearly identical to each other, okay, maybe a single gene or two different between like gorillas and chimps, okay, but if you examine our chromosomes, we have a chromosome, chromosome number 2, that have the genes, the same genes that chimps, gorillas, and orangutans have on their chromosome number 2 and 3. So our chromosome number 2 is a result of their chromosomes sticking together. And not orangutans, gorillas, and chimps chromosomes sticking together, but the ancestor to these guys, or our ancestors' chromosomes, got stuck together. Their chromosome number 2 and 3 got stuck together to result in our chromosome number 2, which is a hybrid chromosome. So not only do we have all the same genes as chimpanzees do on our chromosome number 2 that they do on 2 and 3, you also will find interesting pieces. So if you examine a chromosome, and you should remember this from general biology or your beginning biology course, if you examine a chromosome, at the tips of your chromosome you have a substance or a region that we call telomere. Okay? And telomeres is a long strand of typically adenine that's added to the end of your chromosomes. Okay? And that strand, or that cap, which we call telomere cap, is really just there. It doesn't code for anything. It's just there to prevent the chromosome from unraveling. Okay, so it's protecting the chromosome. So you have telomeres on the tips of your chromosome. In the middle of your chromosome, you have another distinct region, which we call a centromere. The centromere provides a binding spot for microtubules, specifically for the kinetochore that ends or tips a microtubule. It reacts with the centromere on a chromosome, okay, and it attaches to it. So in the middle you should have centromeres, and at the tips you should have telomeres. Our chromosome number 2 has telomeres at the ends, but you also have telomeres in the middle. Okay? And instead of one centromere, we have 2 centromeres, which shows us good evidence that our chromosome number 2 is the combination of our ancestors' chromosome number 2 and 3. Okay? So this is a rearrangement of the number or structure of chromosomes. This happens often. Sometimes it results in mutations when you have too many copies or not enough copies of a chromosome, but sometimes it doesn't really change anything, but it gives you this evidence of an evolutionary event. Okay? The combination of chromosomes. And then most of you know that any really mistake in the genome, so replacing an adenine with a thymine, okay, or an adenine with guanine, or any mutation where you're getting a point mutation and you're switching the nucleic acid or nucleotide that belongs there, okay, or was originated there, you're moving that out, okay, you can get a mutation, and that's a mutation of the existing alleles. Sometimes it works. Sometimes the result is just a small change in the protein structure, which gives you maybe a different reflection of color. And that's how you get different hair colors, eye colors, those kind of things, skin color. It's just a change in the protein shape. Okay? Other times it might not work. Okay, so you get this change in the protein shape, and instead of being something that's important for color, maybe it's important for texture. And now the wing of the organism doesn't develop, okay, or it's misshapen. Okay, or you get a misshapen kidney or a misshapen liver, okay, and they don't function. Okay, so a lot of times mutations are maladaptive, meaning that they just are not good for the organism, but occasionally you get new alleles that pop up and they're beneficial. All right, so genetic variation, when we're talking about a population, is pretty much unlimited. Okay? As long as the population is fairly large and has multiple chromosomes and multiple genes, then for the most part the amount of genetic variation is unlimited, because the amount of crossing over that occurs is not predictable. The amount of independent assortment, or how the chromosomes assort themselves, is not predictable. When mutations pop up and arise, and when they're good, when they're bad, not predictable. So you have a lot of these things. Whether or not chromosomes stick together or rearrange themselves, okay, again, not predictable. So you have a lot of variation that can occur just from meiosis and processes that are related, like synthesizing DNA, things like that. Okay, so since variation is basically unlimited, we have the ability to examine a population to see if that variation is changing, or if that frequency of alleles is changing over time. Okay? So we could take a population and we can examine that population. We can examine their genetics, or we could just examine their phenotypes. Okay? And this is what Hardy and Weinberg did. Okay? But we now have the capability of examining the exact allele differences. Okay? So maybe it's 75% dominant, 25% recessive in the original population. If the next generation, something going on, maybe natural selection took place, maybe sexual selection took place, something happened. If you're moving and you're changing the frequency of alleles, so now instead of having 75% dominant, now you have 71% dominant, okay, you have a population that is going through evolution. It's changing its allele frequency over time. That's the definition of evolution. Okay? It might not result in any physical features that you could tell, or physiological features, or behavior features. Okay? But nonetheless, the allele frequency of the population has changed over time. So the question has been posed, and it was posed by Hardy and Weinberg, okay? Do populations have to be able to evolve? Are they always evolving? Does evolution have to occur? Okay? Now, Charles Darwin, when he came up with natural selection, evolution by means of natural selection, he suggested that populations are always evolving. There's always this pressure from outside sources to change when the outside sources change. So when you get environmental change, when you get climate change, when you get these things, it puts pressure on populations to change. Okay? Now, they're not changing like Lamarckian. They don't get to choose how they change. However, the organisms that survive are chosen by the environment. Okay? So population geneticists really study gene pools or genetic events. They study how populations change over time. So maybe they go out and sample a population, and then they go back and sample the population 5 years later to see if the population has changed within those 5 years. Maybe you had a population that lived in a prairie where there's grasslands and things like that, and then next to that prairie, housing developments came around. Okay? So maybe a population geneticist is interested in, hey, what did the population look like before the housing development? What's the population look like now? Did the population evolve? Did it change given the new environment? Okay? So that really brings us to Hardy-Weinberg theorem. Okay? So Hardy-Weinberg, again, like I said before, they were interested in whether or not populations have to evolve, and are they evolved? Okay? And they noticed that just mixing of alleles, those things that I talked about before, crossing over, independent assortment, chromosome rearrangement, and mutations, it's not really enough to drive a population's allele frequency in one direction or the other. Okay? So just the mixing there is not enough to change it for future generations. Okay? Now, it might be the precursor. It might help, it might start the ball rolling, but it's not enough to drive evolution. Okay? So, if the population's alleles are not changing, if the relative frequency of dominant to recessive does not change from one generation to the next, or from 1 year to 5 years later, then evolution is not occurring. Okay? And Hardy-Weinberg came up with a theorem to describe this. Okay? So there's certain assumptions that Hardy-Weinberg had to make in order for their theorem to be legitimate. Okay? So again, Hardy-Weinberg theorem says that if populations are not changing, the frequency of alleles are not changing, then they're at equilibrium and evolution is not occurring. Okay? So in order for this to occur, Hardy-Weinberg came up with 4 or 5 things. Okay? It depends on what textbook or who's teaching it. Okay, so it's 4 or 5 general things that the population must have. First, population has to be large. Okay? Infinitely large. Okay, I don't want to say that smaller populations can't fit Hardy-Weinberg, but it's more difficult. The larger the population, the better chance that it will fit Hardy-Weinberg equilibrium, okay? Because of something called genetic drift. Okay? Genetic drift, or when gene frequencies change by chance, okay, happens more often in small populations. Okay? It's just like taking a coin and flipping a coin. If you flip it 10 times, the odds that you get 50-50, okay, are still 50-50. That's the odds, right? But the chances that you get 50-50, 5 heads, 5 tails, I urge you to try it. I doubt you're going to get 5 and 5. And so, but if you flipped it 100 times, now your population's 100, you're going to be pretty close to 50 heads, 50 tails. And then you say, okay, my population's 1,000, so I'm going to flip it 1,000 times. Now you're going to be very close to 500 and 500. Much closer than what you were with 10 flips. Okay? So the probability or the frequency change by chance alone is probably going to be greater. Now, the probability doesn't change, but chance does. Okay? All right, number 2. Individuals cannot migrate into or out of the population in order for equilibrium to be met. Now, this should make sense. If you have individuals migrating in, then you have new alleles coming into the population. If you have individuals migrating out, then you have alleles removed from the population. So you can't have that, otherwise you're changing the frequency of alleles in the population. Okay? Mutations cannot occur, okay, or they have to occur at a very small rate, and they're not introducing new alleles. Okay, so a population cannot have these new alleles, coming into the population to change the frequency in any way. Okay? And for Sexual reproduction must be random. Okay, so there's no mate selection. And populations will just reproduce based on, what's available. Males will just mate with females. There's no choice. There's no singing. There's no dancing. There's nothing. No fighting. Just organisms will mate at random, purely at random. Now the fifth one that's sometimes represented is natural selection. Okay? Now, sexual reproduction must be random kind of incorporates natural selection. Because if there's no selection going on, whether it's mate selection or nature selection or artificial selection, then, that should make sense. We know that natural selection, nature selecting for certain individuals, is going to violate Hardy-Weinberg equilibrium. Okay? So sometimes 5 is represented as no natural selection. Other times it's just, kind of a give me. If you have natural selection, which is going to change the allele frequency, then you know that that's going to violate Hardy-Weinberg equilibrium. Okay? All right. So when any of those features, any of those 4 or 5 features are not met, then the population is changing. The allele frequencies are changing, and the evolution is occurring. Okay? So again, Hardy-Weinberg, we're trying to figure out ways in which you could examine a population to determine if it was evolving. If they met all those assumptions, then they were not evolving. The population was not changing. Its frequency, allele frequency, was not changing. It was stable, or it was considered to be at Hardy-Weinberg equilibrium. As you can imagine, most populations on the planet do not fit these assumptions. Okay? Natural selection is occurring, sexual selection is occurring, mutations are occurring, immigration are occurring, populations can be very small in regions. So almost every single population on the planet right now, if you were to examine them, they are evolving, they are changing. Now, some of them might be stable now. They might be at Hardy-Weinberg equilibrium right now. Okay? But based on these characteristics, we know that every population has experienced evolutionary change at some point. Okay? And so you might be able to find a population that meets these assumptions, okay? But give it time. Okay? Because some of those environmental factors, some of the region in which that population resides in will change, and the population then will have to change. Or the mutation rate might change. Or you might have individuals leave the population, okay, which would change the frequency, etc. All right, so that brings me to kind of the final piece. So it wasn't just about developing assumptions for an equilibrium. Hardy-Weinberg also developed mathematical equations to figure out whether or not a population is changing. And so these are really predictive values in which you could examine a population and predict how many of those individuals are homozygote dominant, heterozygote, homozygote recessive, okay? As long as it's simple Mendelian genetics. Okay? A typical monohybrid, one-for-one, genotype to phenotype situation, then Hardy-Weinberg equations can be used. Okay? So let's examine one. Let's say, okay, and you can use all 3 of these equations. Most of the time you're using, okay, this equation and the bottom equation here. Okay, p squared represents how many homozygote dominant individuals, or the frequency of homozygote dominant. 2pq is the frequency of heterozygotes. Q squared is the frequency of homozygote dominant or homozygote recessive individuals. P is dominant, Q is recessive. Okay? So let's say dominant is 0.7. That's the frequency. 70% of the population, whatever the population is dominant. Okay? So you could just stick that into P and solve for Q. So then you would know that the recessive is 30%. But maybe we're not just interested in knowing dominant and recessive. And so we could probably figure this out by just examining a population pretty easily, right? So you go out and you examine, say, pocket mice. Catch a bunch of pocket mice in a population, and you look at them. You look at their coat color. And let's say their coat is brown, and that's dominant, or their coat is like white, and that's recessive. And you come up with these numbers, 70-30. But that might not be enough for you because you're interested in, well, how many of the individuals are actually carrying the recessive trait? Not showing it, but carriers of it. Okay, so we want to know how many are heterozygotes. Okay, so you can just start plugging things in. You could say, all right, so if 0.7 is dominant, we can stick that into the p squared, okay, square the dominant, and that gives us how many homozygote dominant individuals there are. Okay, about 49% of the population are going to be dominant. Okay. We can do the same. We can stick 70 and 30 in here and figure out how many are heterozygote dominant, about 42%. And then maybe we're interested in how many are recessive. Okay. It's about 9%. Okay. And that's how Hardy-Weinberg equilibrium works, or the Hardy-Weinberg equation works, in order to be able to predict how the population is shifting, or what portion of the population is heterozygote, what portion is homozygote recessive, etc. You can also do this the opposite way. Maybe you knew how many were homozygote recessive, okay, and you're trying to get at how many are heterozygotes. Okay, you just take the square root of that. That gives you Q, and then you can solve for P, and then you can solve for this portion. Okay, so there are ways at which you can flip back and forth. Okay, but again, this is simplistic. You'll see that as we progress, most features that we examine don't fit a normal Hardy-Weinberg or a normal Mendelian genetics, and it's kind of difficult to use these. Now, for simplistic factors, and every population will have a set of Mendelian genetics or Mendelian features that fit kind of a monohybrid, simple, one-to-one genotype-phenotype interaction, and you could use that on this. But as you'll see, a lot of features that we're interested in are multiple traits, epistasis, incomplete dominance, codominance, all kinds of things that are going on, gene-by-gene interaction that's going on that would make this not really all that useful. Okay? So next time, we're going to actually talk about how populations can change. How can you change the allele frequency of a population? What are the means by which it can change? And I'll give you some examples of how those, how that change comes about, and some classic examples of evolution by means of natural selection or by genetic drift, gene flow, these kind of things. Okay? So next time we'll hit those things.